last week we discussed the replication, and then we mentioned about this coupling, right? The replisome. Okay, so this is the picture, the cartoon of the replisome. So this is a classic model of the E. coli replisome. Okay, so they have this they have the highly coordinated leading strand and lagging strand synthesis. They couple together. So as you can see that here we have leading strand, we have PO3, the coenzyme, all right, and replication fork, and then we have this helicase, right? So helicase always attached to the lagging strand, right? Lagging strand, so they move from 5' to 3' to open a double helix. And then we have this clamp loader, okay, clamp loader. And also we have polymerase 3. The polymerase 3 is alpha subunit, beta subunit, epsilon subunit, and theta subunit. And then we have clamp loader, and then they tried to put both together, right? So that's a replisome. This is just the cartoon to show you the replisome. And today we continue this replication pattern. Okay, so basically the eukaryotic and bacteria DNA, they replicate bidirectionally, all right? So it doesn't matter whether it's the circular chromosome or linear chromosome. They replicate bidirectionally, and they begin from the place we call it the original replication. Original replication basically are the DNA sequence, okay, for the replication complex to recognize, so they can bind and then start the replication. And also the original replication, also called a replicon. Okay, and on the right side here, you can see this is a typical bacteria chromosome, and this is linear, the eukaryotic chromosome. And then for circular, so for example, the E. coli chromosome, they have a single replicant. Okay, they have a single replicant. That means only one location that shows the original replication. And then the DNA replication begin with the creation of the bubble, and which is a small region that the parental strand have separated and the progeny strand have been synthesized. Okay, like this, open up, right? So that bubble. And then you have two replication forks, one on each side, and then they will proceed to the other end, right? When they proceed, okay, so the bubble will expand, okay, and then so you will see it become like a theta, all right, the theta shape until the end when both replication fork, okay, 1 and 2, when they both meet at, at the end, all right. At the end that we will discuss later is a special sequences, termination sequence here. So they will meet here and then they will separate, right? So you get two chromosomes, right? So that's for the E. coli. And then the eukaryotic cell, okay, they have many replicons. Remember, the eukaryotic genome is very big, so they need to have many replicons so they can perform the replication, all the replication at the same time. Okay, and then again they open up and then they proceed on both sides. So bidirectional replication, that's what this means. Okay. Yes, Marina.
When we're looking at a coli genome, isn't it? How do I put it? Isn't there multiple enzymes working to create more replication forks to increase that turnover rate and basically replicate?
Yeah, many enzymes like this here,
Okay.
all the enzymes.
Because I
All
remember
right.
from microbiology that I think the turnover time of replication was, I think, 20 minutes. So that would show that there's multiple replication forks in um, progress working at one time.
Yeah, the turnover, I mean, every generation, 20, 30 minutes. Yes, that's correct.
Okay.
They will replicate the whole chromosome very quick. Okay. And then the rolling circle replication. Okay. Rolling circle replication is very special. And the you can see this commonly seen in the virus. Okay, like the bacteriophage, the virus. They use the rolling circle replication. Okay, and they have a specific feature of the circle DNA replication. Basically, for example, number one, synthesis of the primer is not necessary, right? Remember when we discussed the synthesis of these replication, we need the RNA primer, we need the primase to synthesize several RNA primer, and then the DNA polymerase 3 will be able to continue the synthesis from the 3' hydroxide group. And for a rolling circle, you do not need the primer, okay? And for the rolling circle, the leading strand is covalently linked to the template. Okay, later on I will show you that the leading strand is covalently linked to the template. And so, for example, when you start this, so basically they will start from this 3' hydroxy group here. So that's why they do not need the prime. And then when they start the replication, all right, when this 5' peel off, all right, open up, so they start a synthesis, 5' to 3'. Therefore, this 5' to 3', the leading strand, linked to this template. All right, that's what it means. The leading strand is covalently linked to the template. And then the DNA replication continue on many run without termination, because as you can see here, the green line, so around this circle, the leading strand. So when they complete the replication, all right, so they remove this already, and then this one, they will synthesize the other strand. And once they finish, then they can continue from here, and then remove this previous strand and continue to synthesize a new strand here. So the DNA replication can continue many rounds without termination. So you can generate multiple copies of DNA, all right? And we normally call this a concatenance, all right? So this is what happened in virus, all right? And then the template strand of the leading strand never separate from the circular part of the molecule. So from the beginning, see the negative here, is serve as a template, right? For the leading strand here, the green one, once this finish, the green one will be released, and then it will start another leading strand. So this template, negative strand, will always there. So that these are the specific unique feature of the rolling circle DNA replication. Okay, so it happened mainly in virus. Okay, so that's a rolling circle replication, and that's why we mentioned this here. And now let's watch a video briefly. Okay.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the.
So that's the reason why for the rolling circle replication does not need the primer, okay?
The intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. A a region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Post-DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method.
Okay, that's a rolling circle replication. And then this is just an example of the RNA virus replication, okay?
Some RNA phage, such as MS2 and Q beta, contain only a single positive plus sense strand of RNA.
So this is just similar to the SARS-CoV-2 virus, because there's also contain the plus RNA, all right?
They infect only F plus strains of E. coli because they attach to the pilus. A positive plus strand of RNA is one that can act directly as messenger RNA. A negative minus strand is complementary to the plus strand and cannot act as messenger RNA. To replicate more positive strands, the positive sense RNA strand must first be used to synthesize an unusual phage-induced RNA polymerase. RNA synthesis from an RNA template does not normally occur inside the uninfected cell, and so the entering plus RNA must code for the synthesis of this RNA polymerase, which is also called RNA replicase. The RNA replicase uses the positive sense RNA as the template for synthesis of negative sense RNA, and the negative sense RNA is used to synthesize more positive strands. The positive strands serve as messenger RNA for synthesis of phage coat proteins and are then incorporated into the phage during assembly. A phage-induced lysozyme lyses the cell, releasing the phage to the environment.
So that's the life cycle of the plus RNA, and this is the example of the bacteriophage, okay? And the SARS-CoV-2 use similar mechanism.
Professor,
So
when you were talking about the plasmid regeneration, so the plasmids are always offering that special antibiotic, usually resistance, special properties, additional genomes, correct?
Can you repeat your question again?
When we were just talking about the plasmids replicating, plasmids are always going to offer that additional genes, such as like that special properties like antibiotic resistance.
Yeah.
Correct?
Yeah.
Okay.
Yeah.
And in the video, it said it started with that RNA primer to reach that the free hydroxyl group to start the synthesis of the new strand, correct?
You mean the rolling circle?
Yes, for the rolling. So
That's a Rep A. That's a Rep A protein. Yeah, Rep A protein. And also it's true that when you are, for example, introduce a plasmid DNA into a bacteria, if that plasmid DNA contain the antibiotic resistant gene, then they can replicate many of these plasmid and also express the antibiotic resistant protein, so that can help the bacteria resist to the antibiotics.
Basically, it's for virulence factors.
Yeah.
So plasmids are always going to replicate through a rolling circle mechanism.
No. For the circular plasmid DNA.
Only circular, so like E. coli.
For bacteria, bacteria, they adopt this method for replication. For virus is a rolling circle replication.
Okay, that makes sense.
Yeah.
Thank you.
Sure. And the termination of DNA replication will occur when the two replication fork, they are moving away from the original replication, and then they meet each other on the opposite side. And here would be a specific DNA sequence that will recognize. We call it the tercide. It's tercide, TER, tercide termination site. It's about a 23 base pair sequence, right? And this termination sequence, they are unidirectional, okay? And so they can be recognized by the unidirectional contra helicase, which is the helicase we call it the terminator, replication terminator protein. It's called TUS. It's a replication terminator protein in E. coli. So they can recognize this 23 base pair sequence, consensus sequence, and then they can prevent the replication fork from proceeding. Okay, so normally in this opposite end here, the third side, they normally have 10 termination site. Okay, and then the binding of the TUS protein, which is the termination protein, the replication terminator protein, they will bind to this third side, so they can prevent the replication fork to pass through. Okay, so that's the termination replication, and then they will separate. Okay, and in general, the mechanism of eukaryotic DNA replication is similar to the prokaryotic DNA replication. Okay, however, there's some more complicated situation for eukaryotic DNA. The number one would be the DNA size, right? The eukaryotic DNA size is much larger than the bacteria. Okay, and also they have a unique linear DNA end structure called a telomere, right? The telomere, right? Remember, you are doing this linear DNA, right? So you have, when the polymerase reach the end, and also when the Okazaki fragment reach the end, there's an issue that we will discuss further, that this end we call terminator, all right? The telomere, the end of the chromosome, telomere, all right? And also the for the eukaryotic cell, the DNA packaging involve the histones, so that when you go through the DNA to replicate the DNA, those histones has to dissociate from the DNA temporarily, and then after the replication fork move past the area, then those histones will come back to package the DNA again. So these are some special issue for the eukaryotic DNA replication. Okay, and of course the most important is the telomere and the histone. All right, because telomere, that's a very complicated situation. When the polymerase reach the end of chromosome, they need to replicate the end of the chromosome, otherwise the chromosome becomes shorter, and have been proved that if the chromosome becomes shorter, and that will influence your lifespan. So that means you will the lifespan will be shorter. All right, and then the histone, because that involves a package of DNA, so histone have to dissociate from the DNA and then come back after replication. So these are very important for the eukaryotic DNA replication. And the first one here, that's the most we focus on, this one is important, is the telomere. All right, okay, so because the eukaryotic chromosome is a linear molecule, and then you have a problem when the replication comes to the end of the DNA. Okay, the synthesis of a legging strand at each end of DNA require a primer, all right, so that the replication can proceed in the 5' to 3' direction. And this become impossible at the end of the DNA, okay, because when you after the legging strand, when you finish the replication and then you remove the RNA primer, then you have a gap, and you cannot fix that. All right, if you don't fix it, then you will lose the DNA, and then the chromosome becomes shorter and shorter after each replication. Okay, so so then we have this special enzyme. Okay, it's called telomerase. All right, so they can recognize the telomere region. The telomerase is a reverse transcriptase. They contain a short stretch of RNA sequence. All right, the short stretch of the RNA sequence normally is AUCCC, AAUC. All right, and so this short stretch of RNA, they can serve as template for the telomere extension. All right, so first of all, you need to extend the telomere. all right? Because here will be the gap after the replication, the lagging strand, then you cannot complete this gap here, all right, after you remove the RNA primer. And now the telomere will come, and then they will replicate they will extend and replicate, and they will extend the telomere sequence, right? The region here, their telomerase, they bind to the 3' end of the leading strand, right? And then they will extend this leading strand so that in the end they have the DNA polymerase alpha, okay? They will be able to synthesize this gap here, right? And later on, they will release the telomerase and then chop off this extra strand, okay? So that's in the telomere. Yes, Mama. Yes, Mama. Mama, I cannot hear you. So basically, the telomerase, they will bind to the telomere, extend this telomere region, all right? And then once they extend it, the DNA polymerase will replicate this sequence here and fill up the gap, and the telomerase will be released, and then this extra DNA will be digested, then you will maintain this DNA here, all right? So you, Marina, yes.
Is this telomere only at like the termination stage of DNA replication, or is this just in generalized?
The telomere, yeah, it will add at the end of the chromosome, which will be almost the end of the termination stage.
So it's
yes.
just about like finishing the DNA replication. This is where this comes intact and basically stabilizes the structure.
Yeah, to finish the DNA replication.
Okay, yeah. And this is only in eukaryotic cells.
Eukaryotic cell, yes, yes, Muhammad. Muhammad, I cannot hear you. All right, okay, so let's move on to watch this video to show you more detail about the replication at the end of a chromosome, right?
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
So that's the replication at telomere, all right? And then based on this knowledge, okay, and then a lot of pharmaceutical companies, they try to design some drug, Okay, so they can inhibit the DNA synthesis, so that they can control, they can treat those bacteria, viral, and the neoplastic disease. Okay, so based on this DNA replication mechanism, and then use this molecular biology knowledge, then you can make the drug. So so basically there are three currently there are three main classes. Number one, the drug that can prevent or reduce the synthesis of precursor, like the nucleotide. And number two would be the drug that can affect the either the template or the priming ability of the growing strand. And number three would be those that can act directly on the polymerase or other enzyme needed for the replication. So all these basically target on the DNA replication. So the goal is to inhibit the DNA replication. Therefore, those bacteria, if you have bacterial infection or viral infection, so those bacteria or viral will not be able to replicate in your body. So that's how you use the molecular biology knowledge to cure disease. Okay. So for example, the first one, the inhibitors. So they say we have inhibitors, right? That can act on the DNA polymerase and also other enzyme of the DNA synthesis. Okay, so we have these the drug like acyclovir, acetylcholine, aminocoumarine, right? So these drugs they inhibit the DNA polymerase activity, all right? So for example, the acyclovir, okay, they can inhibit the DNA polymerase of the HSV, the herpes simplex virus, all right? So that's this drug that can cure can treat the HSV infection, okay? And then the acetylcholine, all right, that can inhibit the DNA polymerase alpha and theta, so they can block the DNA replication. So then, so they can basically they work on those viral DNA polymerase, so they can prevent the viral DNA polymerase activity. Okay. And the aminocoumarine, basically, it's an antibiotic like the novabiocin, all right? So they can inhibit the DNA gyrus, okay? They can inhibit the DNA gyrus. So basically, they work on the bacteria, okay? So these are some examples to show you how you apply the knowledge to prevent the bacteria infection and viral infection, okay? And we will skip this because mitochondrial replication, basically, the mitochondria basically divided by the developing a ring around the organelle that constrict to pinch it into a two half. And the mechanism is similar to the principle of bacterial division, okay? So they combine both the replication and the segregation mechanism together, and then to split the DNA. That's a mitochondrial replication. And the fidelity of the DNA replication. So basically, the precise copy of the genetic information during the DNA replication that can provide the genetic stability to the living organism. Therefore, you have to make sure the replication is accurate, right? And however, the error can be introduced during the replication. And if that error has been introduced, then the cell, they have the mechanism to fix the error. So they have this proofreading activity of the DNA polymerase, right? So they can make sure the replication accuracy. So for example, the 3' to 5' exonuclease activity of DNA polymerase, that one is a key player to make sure you have accurate DNA replication. Okay, it basically is in the alpha subunit of the polymerase 3 holoenzyme. Okay. And the epsilon, the alpha subunit, they catalyze the nucleotide polymerase, but the epsilon subunit of the polymerase 3, they contain the 3' to 5' exonuclease. So the 3' to 5' exonuclease will be able to double check whether the nucleotide they incorporate is complementary to the template or not. If it's not, then the exonuclease will remove the wrong nucleotide. Okay, so that after this proofreading activity, then we will be able to get this accuracy about 10 to the 9, 10 to the 10 to reduce the error to down to the 1 in every 10 to the 9, 10 to the 10 base pair. Okay, so that's the fidelity of DNA replication. And of course, there are sometimes that the mistake that the wrong nucleotide that they escape this proofreading mechanism, and then they will introduce the mutation. Okay, and generally speaking, there are two different category of mutation. One is a large scale, and the other one is small scale mutation. So the large scale mutation would be like the chromosomal mutation. All right. So you have this that's chromosomal mutation. So you have the translocation, inversion, deletion, and non-disjunction of the chromosome. Okay. So these are the large scale mutation. Yeah, Marina.
Would a large scale mutation be considered sickle cell anemia? How instead of, I think, valine is replaced instead of guanine, if I'm correct, or no, glutamine? I just know it's valine is replaced by something. It's one amino acid, and then that creates obviously the sickle cell red blood cell.
Oh, for that mutation, that is a small scale mutation, which we will discuss
That's
in a
a
minute.
small scale considering the
Yeah.
physiological.
That large scale, that's a big piece of the chromosome
Oh,
DNA
okay.
replaced. Okay.
Okay.
Would that be
So, you
considered,
have-
Professor, like a frame shift mutation?
No, those are small scale that we will discuss in a minute.
Okay.
But these large scale, I mean, it's large piece, right? Not only affect it's a very large region. Okay. So later on, we will discuss these large scale mutation. And for the small scale, okay, small scale, that would be what you just mentioned. So small scale, we have the point mutation, so that would be the base substitution.
So this is more when there's one amino acid in place of the other, or something along that line.
Correct, yes.
Where it's not
That's right. That's sickle cell. Sickle cell, that's a one amino acid change,
yes.
right? Because the one nucleotide change, and then you make the whole amino acids change. So that's a small scale. That's a point mutation. So for a point mutation, I mean you change one nucleotide. Okay. Okay. That's a small scale. And then, for example, for the point mutation, we have 3 different types of point mutation. All right. So we have missense mutation. So that means missense mutation, you change one nucleotide from A to C, then you change this amino acid. That's called missense mutation. And then we have nonsense mutation. So that means you change one nucleotide from U to A, then you change this amino acid to the stop codon. So that's nonsense. Also, we have silent mutation. So that means you change the nucleotide, you don't change the amino acid. That's called silent. So we have missense, nonsense, and silent mutation. Okay, so these are the point mutation. And then, it's a base substitution, and the point mutation is base substitution and can also be further divided into the transition and transversion mutation. It depends on the type of the base changes. Okay, so if purine to purine change, we call it a transition. All right, if the purine to pyrimidine or pyrimidine to purine mutation, we call it the transversion. All right. So like A and G, all right, adenine, guanine, that's purine, right? A and G, and C and T, that's pyrimidine. So if it's purine to purine change, so like if you change it A to G, so that's the transition, right? Purine to purine. But if you have the purine to pyrimidine, so if you change it A to C or A to T, then that'll be transversion. Okay, because the purine to pyrimidine. Okay, that's this kind of the base substitution, the point mutation. Okay, and then we also have this the insertion and deletion. All right, insertion deletion that can give you this friendship mutation. Okay, the friendship mutation. So for example, in this case here, we have original base sequence ATG, AA, GAG, UAU, and then you have addition between these two A. So now it becomes ATG, AAG, and then move this to next reading friend. So it'll be AGA and GUA. So then that'll be the friend shift, right? You shift the reading friend. Or you can have base deletion, you delete one nucleotide, and also you shift the reading friend, right? And so if you add one or two or delete one or two nucleotide, that can cause the friend shift. And for the 3 base insertion or deletion, sometimes they don't change the codon frame for protein synthesis, but they may cause diseases, all right? So if you add 3 nucleotides, let's say if you add 3 nucleotides in between, you don't change the reading frame, but the overall protein sequence has been changed, all right? So that's what it says. 3 base insertion or deletion do not change the codon frame for protein synthesis, but may cause a disease. So this is an example of a disease that when you add 3 nucleotides, all right? So you have these 3 nucleotides. So that may cause the Fragile X syndrome. Okay, Huntington disease, myotonic dystrophy. All right, so all these diseases, the 3 base, 3 nucleotide addition. All right, so that will cause the disease. And then also some other different kind of mutation is the chemical stability. So for example, yeah, what's the question? Let me see. So this is just the repeat of the sequence, right? CGG. If it's the repeat of a sequence 5 to 54 times, there is no inference, all right? But if it's 60 to 230, then that'll become the Fragile X syndrome carrier, all right? Then if you have 230, you repeat this 230 times and more, then that will show Fragile X syndrome. That's what this means. For Huntington, the repeat sequence CAG, 11 to 30 repeat. If you repeat 11 times, then it's just wild type, okay, no disease. If you repeat 36 times of CAG in the chromosome, then that will show Huntington disease. That's what this means, okay? The trinucleotide repeat.
Oh, so Professor, so you have to repeat those nucleotides multiple times for the disease to be effective.
Correct. So for Fragile X, yeah, at least 230 times. Okay, Huntington, that's 36 times. Okay, so that's what this means. And the tautomeric shift. So basically, that's the chemical structure of these nucleotides. Okay, so basically, for example, adenine and cytosine, right? Adenine and cytosine. Okay, adenine and cytosine. They basically, they mainly they use the amino, this amino configuration. Okay, sometimes they can have immuno configuration, but when they change to this immuno configuration, then their base pairing will no longer perfect complementary. Okay, and the guanine and cytosine, right? Guanine and cytosine, they normally adopt the keto configuration, right? The keto, this column here, these two keto, and adenine, cytosine as amino configuration, and guanine, thymine, the keto, and sometimes they can shift to the eno configuration. Once they shift to this different configuration, then we call, there will be the tautomerization, all right? Tautomerization, okay, when they form the complementary sequence. So like, for example, in this, during the replication, okay, during the replication, if this A, right, shift from this amino configuration to immuno configuration, then we call this as tautomer, all right, tautomer. Then they will form, this tautomer A will form a complementary sequence with C instead of T, all right, okay. And if we have tautomer C, so here tautomer A, then you introduce the C, and then for the next cell division, then this C will match with G, right? So that'll be the transition mutation. So you introduce this mutation, okay? So that's a tautomery shift to introduce the mutation, okay? So that's what this means, the tautomerization of base, okay? And that's how they also, one of the mechanism they can introduce the mutation. And the mismatch, okay, so if they escape mismatch,
Professor, was that
yes,
picture showing the base pair go to an A to a G? It's hard to, it's really small, I can't see it. So it's going from an A to G at the bottom?
so here's yes, here's A, all right, A, right, is a tautomer A, right? So A, instead of this configuration, this tautomer A is this immuno configuration. So this A will complementary to the C, all right? So they introduce the C here. And now for the next replication, this C,
Oh, I see.
then
So now the C
bring
goes
in
on
the
the
G.
other side, and then the complementary base will go on the
Yeah,
opposite. Okay, I see.
okay.
Also,
So
Marina,
this,
you can zoom in on your left-hand side. You see where it says show view controls? You're able to
oh,
zoom in.
thank you. Oh, that's nice. Okay, thank you.
You're welcome.
And the mismatch, okay? So mismatch repair. So that's when the mutation escape the proofreading, right? Proofreading. Then we have several mechanisms to repair the mismatch, because the that can because if you carry this mismatch, that bring in the mutation, okay, that bring in the mutation. And so the mismatch is one of the excision repair system. There are three types of excision repair system. One is the mismatch repair, and second one is the base excision repair, and third one is the nucleotide excision repair. Okay, and this one here, mismatch repair, that can fix the problem that if you that we just discussed here. These are point mutation, right? So for this mismatch repair, basically we need the three key components. So we need the mut S, mut L, and mut H3 proteins. Okay, these are the three mismatch protein. Okay, they will basically the mut S. All right, the mut S, the protein will scan the DNA sequence and bind to the mismatched base. All right, bind to the mismatched base. And how do they know which one is the parental strand, which one is total strand? It's about this. The methylated strand, all right? Methylated strand, so that the mute S will know the strand carry the methylation will be a parental. If they find mismatch and they know this one is the daughter strand, all right? So they know this one is the daughter strand. So they will bind to the mismatched base, all right? Then the mute H protein, all right, will recognize the methylated DNA sequence of GATC, all right, the GATC on the parental strand, all right, and then mute L will link the mute H and the mute S together, and then the mute H protein will digest the daughter strand, okay, daughter strand, all right, and then the helices UVRD, all right, UVRD will unwind the DNA from the nick and then proceed past the mismatched nucleotide. Then exonuclease will cut away the nick strand, and then the single strand binding protein stabilize the resulting single strand. Then the excised DNA region can be resynthesized by DNA polymerase 3, and then DNA ligase finally seal the nick to form a double-stranded DNA. So that's the mismatch repair. Okay, so let's watch the video. Okay.
This is a video of a 3D animation on the mismatch repair pathway in E. coli. In the cytoplasm is the E. coli's DNA, the molecule that contains all of its genetic information. DNA is made up of nucleotides, each of which contains a phosphate sugar backbone and an organic base. The two strands of DNA are held together by hydrogen bonding between complementary base pairs. Adenine pairs with thymine and cytosine pairs with guanine. DNA mismatches occur during DNA replication. For simplicity, this animation will show DNA as a flat molecule. Before replication can begin, a helicase needs to unwind the DNA molecule. In E. coli, DNA polymerase 3 catalyzes the incorporation of new complementary bases to the new DNA strand. This is only the alpha and beta units of the DNA polymerase 3 enzyme. However, it is possible for DNA polymerase 3 to incorporate a non-complementary base to the new strand. This is known as a mismatch. A mismatch could result in a faulty protein being coded for, so it needs to be repaired. The first step in the mismatch repair pathway in E. coli is recognition of the mismatch by the mute S homodimer. Mute S recognizes the mismatch and binds to it. Then, the mute L homodimer binds to mute S, ready for a later step in the mismatch repair pathway. Meanwhile, up to a kilobase away, the mute H protein recognizes a GATC site. Mute H is a weak endonuclease, which will create a backbone incision between the guanine and adenine residues on the newly synthesized strand when activated. Mute H is able to differentiate between the newly synthesized strand and the template strand by the lack of methylation markers on the newly synthesized strand. This is why the pathway is also known as methyl-directed mismatch repair. In order for mute H to become activated, it needs to bind with mute L. This causes the DNA to bend. The newly formed backbone incision allows an exonuclease to enter and begin excising nucleotides from the newly synthesized strand. The exonuclease excises the mismatched nucleotide and stops excising just beyond it. Now, DNA polymerase 3 can begin to resynthesize the new strand, inserting the correct complementary base where the mismatch used to be. The final step is for DNA ligase to ligate the backbone nick. The mismatch pathway is now completed, and all the newly synthesized DNA is complementary to the template strand.
All right, so that's mismatch repair.
Question, Professor, I just wanted to clarify on the video. Which one was methylated? The daughter or the original?
Parent.
The parent. The parent is
Yes.
not methylated?
No, parent is methylated.
Methylated. So is that the one that's being excised or no?
No, that the daughter been excised.
Okay, so the daughter gets excised, but the parent stays.
Stay.
Stays. Okay.
Yes. Yes. Mahama. I cannot hear you. Jessica.
I just have a question on
Yeah.
on like the methylation. So like is it because the mute H like that's on the parent strand like is and like because that's methylated, is that why it bends, or like it just bends because it needs?
They need to bend it so they can recognize that's the region they're going to fix it. Yeah, and so that's why they link it together. So basically, the mute S
So it
they will link together
I'm
to
sorry,
bend it.
you can continue.
They bend it
So
that
it's
they can
mute S linking.
H and L.
So it's mute S but connecting H and L. Oh,
Yeah. Basically, it's a L, mute L link the H and S together. And so they bend the DNA. So the
okay.
DNA ligase, yes, sure, no problem, Jessica. So the DNA ligase always to repair the phosphatized bond. Okay. Marina.
What is the probability of these mutations happening? Like, what is the physical statistics for the mutations?
Mutation depends. I mean, normally it's like a 1 in 10 to the 9.
Mm-hmm. And now,
Yeah.
what's like the statistics for it being repaired versus non-repaired? Like, is there ever a chance
Repair would be
where it's not repaired?
not so not re, so basically not repair. So 10 to the 9 1. So with the repair that you can lower the probability even a 10 to the 2,
Okay.
lower 10 to the 2, even further. Yeah.
But there's always some slim chance that these mutations will actually never get physically fixed.
Yeah, if it's not been fixed, then you become the mutation.
And then
Right.
either we can have the silent where we don't know even, or it can be problematic, resulting in any diseases, pathological conditions, and so forth.
Right. So
Okay.
one obvious example would be the for example the SARS-CoV-2 virus. All right. So SARS-CoV-2 virus. So you can see so many variants, and those RNA virus, they normally they have this error during the replication, and most of the time their proofreading mechanism and also DNA repair system not as accurate as the eukaryotic DNA. So that's why they can mutate very quickly. And that's also the reason why, for example, every year you need to take the flu shot.
The flu shot
Because,
with
yeah,
the proteins, the HA and NA.
those viruses, that's why they get the mutation very quickly.
So now that you bring that up, now that we're dealing with this pandemic with COVID, is there even, do you think there's a chance of us controlling the mutations?
No, you would never be able to control the mutation because the virus, when they are doing the replication, and their proofreading mechanism, those repair mechanism is not as good as eukaryotic cell. So that's how they survive because they
So,
need to mutate, and then those mutant, if they can fit, then they can stay. If they don't fit, then they will disappear.
okay, I
That's one
understand.
of their reason mechanism they can survive. Yeah.
So basically, the mutations that are going to result are going to be less virulent, have less
Will
virulent
continue.
factors.
It will be continue. You will continue. Yeah.
Okay.
That mutation will continue.
They will
However,
continue, but they're not going to be as devastating the effects when getting the disease.
It depends on where the mutation is. If it let's say the mutation
whether the
has
mutation, yeah,
an
can
S
fit.
protein, then it's gonna cause more it to be more infectious. So then it's gonna even mutate even more. It depends on where the mutation is.
Yes, depend on the location of the mutation. So, so if the mutation, later on we will discuss the SARS-CoV-2 genome, and then you will see because a different region they're responsible for different they have different function, and
Okay.
then for example, if they mutate at the specific location that's responsible for the recognition of the ACE2 receptor, so perhaps that enhance the binding activity or maybe lower the binding activity. And whatever the mutation, as long as that can enhance the virus activity or ability to survive, that virus will survive. That
Okay, I see.
mutation will survive. Yeah.
Thank you.
Okay. So the DNA damage. All right. So basically, the organisms are frequently exposed to mutagens, and the mutagen that can cause the damage to DNA. Okay. So we have the physical agent and the chemical reagent. All right. That can cause a mutation. All these we call both the physical agent or chemical reagent. As long as they cause mutation, we call them mutagen. Right. And this including the environmental chemicals, ionizing radiation. Okay. And the process that produces a mutation is called a mutagenesis. Okay. And if the DNA damage is left unrepaired, then that lead to the mutation. Okay. And the base analog. Basically, these are the mutagenic chemical that can substitute for the normal purine or pyrimidine during the nucleic acid biosynthesis. All right. Once they replace the normal purine or pyrimidine, then they introduce the mutation. All right. So for example, this one here, we have a thymine, right? This is structural similarity. This is a 5' bromouracil. Okay. We have a ketophone and the enophone, right? So the ketophone here, right? They form a complementary with the adenine, but the enophone here, this one will form the complementary sequence with guanine. So if the thymine has been replaced by either of these base analog, if they say replaced by the ketophone, then it's okay. It's still a match with the AT complementary with A, right? If this one is replaced by this analog, right? 5' bromouracil enophone, then they will introduce this guanine. Okay. So this is just to show the analog can also introduce the mutation. And then alkylating agents. Alkylation basically is a process where the electrophile attack the negative charge of DNA molecule and then add the carbon containing group, okay, to the chemical, all right, the alkyl group. So just like here, we have the guanine cytosine, all right, And now this place, right? This place been replaced with this archaeal group here, and this process alkylation, all right? Alkylation. So if this one has been replaced, right? So they added EMS, which is the mesansulfonate. It's one of the alkylating agent. So they will alkylate the O6, okay, oxygen 6 of the guanine, right? Oxygen 6 of the guanine. And then they will transfer the ethyl, which is CH3, CH2, that's the ethyl group, all right, to the oxygen, the O6 of the guanine, all right? So you have this group here, the ethyl group. And then they will not be able to complement with the cytosine anymore. Then instead, they form a complementary sequence with thymine, okay? And this is also another pathway to introduce the mutation. So the large scale, I think it's just affect this single nucleotide, and then that will introduce this single base substitution. So from the base substitution, then that can lead to a 3 possibility. The 3 possibility, okay? No, we are not talking about changing the pH. The function of protein. So that's why what I try to say now. So once you introduce one base substitution, then there are 3 possible results. One is nonsense. Remember nonsense? Here, missense, nonsense. Missense, nonsense, silent. 3 possibility. If it's a silent, then they don't change the protein. All right. If it's nonsense, then they change it to stop codon. If it's missense, they change the protein completely. So that's the 3 possible results. If you introduce this archaeo, the ethyl group, okay.
Oh, it's introducing a alternative group, and that's what's causing the mutation.
The mutation is because now they match with T instead of C.
Oh, I see. Okay.
All right.
Okay, I saw alkylating, and I was thinking of like relative of pH. So I.
No. It's nothing to do with that.
Okay, it's just the group.
Yes, the chemical group. Okay. And then so the UV radiation, basically UV radiation will cross-link the this you can see here the cross-link the adjacent pyrimidine on the same DNA strand, so they can form the 2 major lesions. All right, and then they form this cyclobutyrene, right? Cyclobutyrene. Okay, and then they form this pyrimidine dimer. Okay, the pyrimidine dimer. So that's the UV radiation. And then ionizing radiation, so that include the gamma ray and X-ray. They interfere with the DNA molecule, okay, similar to the UV, okay. And then
So
yes.
I'm sorry, no, continue. I think my mic is working now. Can you hear
Okay.
me?
Yeah, I can hear you now because yeah, before it didn't work, and now
Yeah.
it works. And then unique things for the ionization is the to ionize the molecules surrounding the DNA, especially the water. Okay, that's because when you ionize the water, that forms the free radical. Okay, which is extremely reactive, and then they can attack the DNA molecule, and then they change the base, or they cause a single double-stranded break. Okay, that's ionizing radiation. And then byproduct damage. So byproduct damage, so the commonly seen byproduct damage will be the oxidation byproduct, which is this 8-oxoguanine. 8-oxoguanine, so we also call it oxo G. So that DNA will suffer Also, they can suffer from the byproduct of the normal cellular process. Okay, so like for example, the superoxides, hydroxy radical, hydrogen peroxide. Okay, so these also can cause a damage, DNA damage. And then for those DNA damage, right? So we have a repair mechanism. So we have single strand damage repair or the double strand damage repair. And for single stranded DNA damage repair, yes, Muhammad.
Sorry. So alkylation is a form of damage.
Alkylation is another damage. They introduce as a carbon-containing group to the nucleic nitrogenous base. In this example, if you treat the DNA with the EMS, which is the isomethan sulfonate, then they will transfer this azo group CH3CH2 to the oxygen 6 of the guanine. Once the azo group being transferred to here, then this guanine no longer complementary to the cytosine. The new one, this one, we call it O6, the isoguanine, they complementary to the thymine. And this is how they introduce the mutation because the nucleotide has been changed. This is not a damage, okay?
Oh, okay. So it's mutation that's not related to free
Damage.
radical or outside. Okay.
No, okay. And so for the repair, damage repair, we have direct reversal of DNA damage. So basically, this is undo the damage. And then we have excision repair. Remember, we have 3 different excision repair. We just mentioned one is mismatch repair, okay? And also the 2 other one is called base excision repair, and the other one is called nucleotide excision repair. And also we have the post-replication repair, all right? And these 3 are the one that cell can use to repair the single-strand DNA damage. Okay, this is just right, undo the damage. The first one, the reverse. Any question? No, direct reversal,
No,
right?
Professor.
Okay, direct reversal. So like this, the thymine dimer, right? You exposed to the UV, right? Exposed to UV, then the pyrimidine, they form this dimer, right? Form this dimer. And then we need the enzyme to reverse this dimer, right? Reverse this dimer. So basically, the mechanism is called photoreactivation because this is a light-induced enzymatic cleavage of the thymine dimer, so that you can cleave the dimer and then to yield to a thymine monomer. Okay, it requires the presence of light, so that's why it's called light reaction. It's accomplished by the enzyme called photolysase, which an enzyme will act on the dimer, right? And the photolysase basically can be activated by absorbing the energy from the light. So that's why it's a light reaction, okay? And then so the photolysase can restore the pyrimidine to the original independent state. And then for the alkylation, all right, the alkylation, right? Remember this one, alkylation, right? Alkylation by the EMS, all right? Alkylation of O6 isoguanine. We need a suicide enzyme, okay? That's the alkylation of oxygen 6 of guanine that can be reversed by this O6 oxygen 6 mesoguanine methyltransferase. This is a suicide enzyme because they will transfer this meso group to the enzyme, and then the enzyme become inactive. So that's why it's called suicide enzyme, right? And this can reverse the damage, right? Reverse the mutation. So you remove the meso group from the guanine.
All right, so just to clarify, so that enzyme removes the methyl group from the DNA, and that's how it repairs itself?
Yes, repair. Yeah, they remove the meso group from the nitrogenous base. Okay. And then the second one, all right, that's first one, direct reversal. Second one, excision repair. Okay. So the first one is called a base excision repair. Base excision repair, right? Is one of the mismatch repair. Okay. So basically, you need the several enzyme. Okay. First of all, if you have one nucleotide damage, and then the first reaction will be to remove this nitrogenase base. Remove this nitrogenase base and then remove this sugar phosphor backbone. And then you synthesize this nucleotide and repair the phosphodiester bond. So the first step, we need a DNA glycosylase, right? They come in here, remove this damaged nitrogenase base, okay? Once this is removed, then we have AP endonuclease come to remove this sugar phosphate backbone, all right? Okay, once this is removed, then the DNA polymerase come in to fill in the gap, and finally DNA ligase repair the phosphodiester bond. So that involves one, DNA glycosylase to remove the nitrogenase base, two, AP endonuclease to remove the sugar phosphate, three, DNA polymerase to fill in the gap, four, DNA ligase to repair the phosphodiester bond. Okay, that's a base excision repair. Let's watch the video quickly.
Some damaged DNA bases are repaired by a system called base excision repair. Base excision repair works differently from nucleotide excision repair, which clips out a section of DNA containing a pyrimidine dimer or other defect.
This is what we are going to discuss in the next section as the nucleotide excision repair, because they remove a piece of DNA, all right? Base excision is just a target to the single one here.
To replace it with new DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil. Uracil can also be incorporated erroneously into a strand during DNA synthesis. If uracil forms in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine, uracil forms a base pair with adenine. During DNA synthesis, DNA polymerase would incorporate an incorrect nucleotide into the growing strand. A few other types of damage are also repaired by the base excision repair system. For example, when adenine becomes deaminated, it forms an unnatural base called hypoxanthine. A mutagen can also damage adenine by alkylating it, converting adenine to 3-methyladenine. Normally, adenine forms a base pair with thymine, but hypoxanthine
Wait, stop.
in its place is mutagenic because it forms a base pair with cytosine. 3-methyladenine is even more damaging. When it is encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair employs a battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA glycosylase, 3-methyladenine DNA glycosylase, and uracil DNA glycosylase recognize hypoxanthine and 3-methyladenine and uracil respectively when these bases are present in DNA. The glycosylases cleave the bond connecting the damaged base to deoxyribose in the phosphodiester backbone. The result is an intact phosphodiester backbone missing a base. The site is called an AP site. In this case, it is missing a purine and is therefore an apurinic site. If it were missing a pyrimidine, it would be an apyrimidinic site. The next step in base excision repair involves AP endonucleases that specifically cleave the phosphodiester backbone at AP sites. The 3' end produced by the AP endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the AP site. DNA ligase then seals the remaining nick, and the repair process is complete.
Okay, so that's the base excision repair, all right? Yeah, Mohammed.
Yeah. So, oh my God. Hello.
Yes,
Hello?
I'm here.
Okay. So is there a chance that DNA polymerase I will make another mistake while it repairs, or no?
There's always a probability to make the mistake.
Oh, okay.
But most likely will not make mistake because it's only one nucleotide. Because the
but it
DNA
cuts other
polymerase
ones too.
they normally make the mistake probably one error every 10 to the 6 nucleotide.
Oh,
Yeah.
okay. Yeah. Thank you.
Yeah. Most likely will not make the mistake, but of course there's always possibility. Okay.
Yeah. Okay. Thank you.
Good. And then the next one, excision repair, is called nucleotide excision repair. Okay, the nucleotide excision repair basically is common for the DNA lesion caused by the UV radiation, oxidative chemicals, reactive oxygen species, cross-linking agent, and the intercollecting drug. Okay, when you distort the DNA structure, if the DNA structure is distorted, then most likely they will use the nucleotide excision repair. Okay, because they can recognize the strand with the bucket damage, right? They have to recognize this bucket damage. And then in the basically in the E. coli, so normally the UVRA, right? The UVRA dimer, they recognize the DNA damage and bind to the damaged site with the UVRB, right? With the UVRB, all right, in an ATP dependent manner, right? And then the DNA strand at the site of damage then will be separated by the UVRB, okay? Because UVRB carry the helicase activity. And then the UVRB can stably associate with the DNA. Then the UVRA will be dissociated. So UVRA recognize, the A recognize the damage area, and B come in, all right? B come in, and then open up the double helix. In the meantime, A dissociate from the DNA, all right? And then the UVRC will come. The UVRC has endonuclease activity, okay? Is recruited to this complex, and then they will cut this piece of DNA, and then DNA polymerase I then will fill in the gap, okay? All right, and here the UVRD basically, the UVRC is to cut this damaged strand, and UVRD it help to release this stretch of DNA, and find the DNA polymerase I come to fill in the gap. Okay, so let's watch this video quickly.
Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
So that's an NER, right? So the direct reversal and the excision repair, they are a true repair process because they remove the defective DNA entirely. And then the cells, sometimes they don't eliminate the defective DNA entirely. They simply just avoid and just avoid that damaged area. And so it's called a damage bypass mechanism or the damage tolerance system, okay? So in this example here, the damage bypass mechanism through the recombination repair, okay? So the recombination repair is also called a post-replication repair because it can repair the pyrimidine dimer that passed the replication. And in this figure here, you can see we have pyrimidine dimer, all right? And then the replication machinery basically just skip this area, right? So that you have a gap. And then in order to fill the gap, then they perform the recombination, right? Recombination with this sister chromatid. And then they move this DNA up here so that now you have complete strand. And then this gap then can be filled in by the DNA polymerase again, because you have the template. Okay, that's called recombination repair. And this is just a very special error-prone repair because that involves this SOS response. Okay, so that's when the bacteria subject to the extreme condition, a lot of DNA damage occur, so that they have this SOS response, okay, to fix it. And basically rely on the proteins called LEX-A, L-E-X, LEX-A protein. LEX-A protein, it's a repressor, okay? So for example, so in the response, the REC-A, which is REC-A coprotease activity is activated by the UV damage, okay? And then followed by the cleavage or transcription repressor, which is the LEX-A repressor. And that will cause the inactivation of LEX-A protein. As a result, the DNA polymerase 5 is formed and become active. And then the POL5 will employ the error-prone bypass of the lesion by synthesizing the nucleotide across from the DNA lesion, even though the damaged region cannot be read correctly. Okay, so that's the error-prone bypass. And then another two mechanism that we double-stranded break repair. Okay, one is the homologous recombination repair, all right, HR, homologous recombination repair, and the other one is a non-homologous end joint, NHEJ. Okay, so these two mechanisms to repair the double-stranded break. Okay, so for the HR, the homologous recombination, they require an undamaged template molecule that contain a homologous DNA sequence that typically on the sister chromatin in the chromatin in the S phase or G2 phase of the cell cycle, so they can perform the recombination repair. And non-homologous rejoin of two double-strand DNA and that occur in all the cell cycle phases. Okay. So for non-homologous join, they just need these two proteins, the Q70, 80, all right, they bind to the broken end and rejoin. That's non-homologous end join. Okay. And then for the homologous recombination, so in the E. coli, basically it's performed by this reg BCD homologous recombination pathway. Okay, basically the reg BCD protein bind to the DNA double helix break and use the DNA helix activity to unwind the DNA toward the so-called the chi site, right? They bind and then they cleave it to the chi site. And then the reg ABCD protein, they can produce a single strand tail, and then this tail will be coated by the reg A protein. And so that can allow the tail to invade a double strand DNA duplex, search for the region of homology. And then they will create a displacement D loop. And to perform this D loop, once the tail find a homologous region, then they will have a nick in the D loop DNA. All right, and then the nick will allow the reg A and the reg A protein and single strand binding protein to create a new tail so they can pair with the gap in other DNA, and the DNA ligase can seal both nick to generate a holiday junction, and also it's called a half chiasmus and the chi structure, and so that they can repair, they can have the recombination repair that double strand break. So that's two mechanism for the double strand break. Okay, so that'll be for chapter 2. Any question?